Hello everyone, welcome to Tutorials by Ayushi. Now this is the 8th video of the course of photosynthesis. In the previous videos I've already talked about light reaction and dark reaction. I've already covered all the processes involved in light reaction, including from light absorption to cyclic photophosphorylation, and I've already talked about how assimilatory powers in form of ATP and NADPH2 are being formed, which are further utilized in dark reaction. In the previous videos I've also mainly devoted a single videos on Kelvin cycle and C2 cycle or photorespiration. Now coming to C4 cycle. Now in the previous video I've already talked about the anomalous behavior that is being shown by Rubisco. Now what happens to Rubisco is that Rubisco in presence of carbon dioxide undergoes carboxylation, that is it combines with carbon dioxide to form PGA. That is the first product that is being formed in C3 cycle. But what happens during two conditions, which is at high temperature, high oxygen concentration, is that Rubisco, in spite of combining with carbon dioxide, it starts combining with oxygen, and that is why it starts behaving as Rubisco oxygenase. Now as Rubisco behaves as a Rubisco oxygenase, it combines with oxygen and it gives out carbon dioxide. Now this is what happens in photorespiration, and photorespiration is basically shown by plants which are present in temperate regions, or mainly monocots including maize, sorghum, sugarcane, euphorbia, chenopodium, etc. Now the plants which show photorespiration also show few adaptations so that they can avoid this energy consuming process of photorespiration. Now what are the various adaptations that are undergone by these monocot plants to avoid photorespiration? The first adaptation includes the presence of dimorphic chloroplast. Now what is it that helps with the presence of dimorphic chloroplast? Now what exactly is dimorphic chloroplast first? Now dimorphic chloroplast means that in a single leaf or in a single plant there is presence of two types of chloroplast, which is granule and agranule. Granule chloroplasts are basically those chloroplasts which consist of grana and stroma both, as you can see in the figure shown below, that in a chloroplast stroma and thylakoids, and the thylakoids are being stocked in form of grana. So therefore grana and stroma both are present in a single chloroplast, whereas in agranule chloroplast only stroma are present and no thylakoids and no grana is present. Now what is it that helps with the presence of granule and agranule chloroplast? Now if you can actually visualize that in the structure of a leaf, the mesophyll cells actually contains granule chloroplast. Now granule chloroplast basically undergoes light reaction or non-cyclic photophosphorylation, and therefore in these granule chloroplast light reaction takes place and assimilatory powers in form of ATP and NADPH2 are formed. Further, these assimilatory powers are further transferred into bundle sheath cells where a granule chloroplast is present, and since the temperature would be low in the lower regions of the leaf and also the concentration of oxygen would be low in the lower region, therefore in bundle sheath cells the basic Kelvin cycle or C3 cycle takes place, where carbon dioxide is took inside the bundle sheath cell and glucose is being formed by Kelvin cycle. Now what actually happens in C4 cycle or Hatch-Slack cycle? Carbon dioxide, now basically two cells are involved in C4 cycle, which is mesophyll cells and bundle sheath cells. Now what happens in mesophyll cells first? Now basically in mesophyll cells granule chloroplasts are present where light reaction takes place, and in bundle sheath cells basic C3 cycle takes place. Now what happens is, in the mesophyll cells carbon dioxide enters the mesophyll cells and it combines with PEP or phosphoenol pyruvate in presence of PEP carboxylase. Now once carbon dioxide reacts with PEP in presence of PEP carboxylase, it forms oxaloacetic acid. Also, I would like to add here is that carbon dioxide do not combine with phosphoenol pyruvate in its carbon dioxide form. What happens is when carbon dioxide comes inside the mesophyll cells, it combines with water and form carbonic acids, and those carbonic acids are what reacts with phosphoenol pyruvate and in presence of PEP carboxylase and forms oxaloacetic acid. Further from oxaloacetic acid, malate is formed in the mesophyll cells, and once malate are being synthesized in the mesophyll cells, these malate then enters the bundle sheath cells, which are in the lower regions of the leaves. Now what happens is that from malate, one carbon is being lost in form of carbon dioxide, and malate converts into pyruvate by loss of carbon dioxide. Now the thing to understand here is that once carbon dioxide is being released in the bundle sheath cells, again carbon dioxide is accepted by ribulose biphosphate in presence of rubisco, where rubisco acts as ribulose carboxylase as it is attaching or as it is combining with carbon dioxide, and which this helps in undergoing of C3 or Kelvin cycle within the bundle sheath cells, and sugars are being formed from C3 cycle. Now what happens with the pyruvate after malate releases carbon dioxide and forms pyruvate here in bundle sheath cells? The pyruvate formed in the bundle sheath cells further moves back to the mesophyll cells, where it is converted into PEP phosphoenol pyruvate again, which is again in presence of PEP carboxylase, which again combines with carbon dioxide and forms oxaloacetic acid, and in this way the complete cycle is undergone again. So I hope you're clear with the adaptations that is being shown by C4 plants or monocot plants to be very specific, and I hope you're clear up with the hatch and sack cycle and C4 cycle. If you like the video, please do follow and also share the video. Thank you so much.